Good morning, everyone. It's a great pleasure to be here with all of you today. And it's an honor to have Professor Antonio Pinheiro with us. He's a distinguished dentist, researcher, and one of the world's leading names in laser therapy and biophotonics. Professor Pinheiro is a pioneer. Since the 90s, I think that is 90s, but
Yes.
he has helped shape the field with extraordinary dedication and continues to be deeply active in both research and clinical practice in the laser field. He is full professor at the School of Dentistry of Federal University of Bahia and head of its Laser Center, editor of Journal of Photochemistry and Photobiology B, currently coordinator of the postgraduate program or graduate program in biotechnology at UFBA, and vice coordinator of the Renor Bio Bahia biotechnology doctoral program. So, Professor Pinheiro, we have been talking a lot about laser, and always when we start to talk about the history of biophotonic in the world, and especially in Brazil, your name is always mentioned. And last week, Professor Marcia Marques made a true mention you and your group in different moments. So thank you so much for
Thank
being
you.
part of our INCT. For us to is an honor to have you and your team with us. So the floor is yours. Marcelo will conduct the discussion with the group today. Uh, and thank you for everything you have been doing for dentistry, for science, and for our group.
Thank you very much, Manuela, for the kind words. It is a pleasure make part of this very selective group of researchers in the field of biophotonics. Well, uh, today I choose to present you a very interesting study we carried out initially with one objective. And from the results of this study, we moved forward. I will tell you during the presentation what happened with that. Today, I will talk to you about the influence of produced water and light irradiation on the composition of exopolysaccharides from a microorganism called Leotia amnesia. Well, it, as I am not seeing you, if this stops or anything, just let me know, Marcelo.
Sorry,
If
just
it stops, because I'm not seeing you, I'm seeing the
oh
presentation. If you have any problem, you just give me a buzz.
no, we can see your screen, but not full screen, only the PowerPoint presentation.
Yeah,
So
if there is any problem, just let
okay,
me know.
I will tell you. Yes.
Yeah. Well, initially, it's important to understand what is produced water. Produced water is a major oil industry byproduct, contains complex salts and organic matter. What's happened in this situation is that when we are removing petrol from the uh, the soil, the deposits are very deep, and for millions of years it is there. One thing that they have also mixed on the the field is water with the oil. And then when you remove the oil, you need to put pressure. And then what the industry makes is to inject water on the field, making increasing pressure and expelling water and petrol. And then this water is separated from the oil, and it goes to tanks and then reinjected within the field. And then this is because it produces a lot of problems in terms of pollution. Exopolysaccharides are high-value biopolymers used in enhanced oil recovery. That is the terminology used in the petrol industry. And the importance of try to use these byproducts, that is the produced water, is to make it a circular economy. It means converting produced water waste into high-value bacterial metabolites that are the exopolysaccharides. The objective of the study was to assess chemical changes on the EPS grown in dialyzed produced water and distilled water, that is the normal cheap normal water, no polluted like it, and also in
Professor,
Oi,
excuse me.
sorry.
We keep you on your first slide.
Wow,
So I
let's
guess you
see.
are already on slide number 3, probably.
Yeah,
So
it's not
it's not,
moving.
no, we are still in the first one.
Strange.
Or at least for me, I don't know. Valeska
Here,
is saying,
his is working.
It's only at the first slide. It's not going forward.
Hold on, I'll check it again. Move.
Stop sharing and start again. Maybe this will help.
Hold on. It's difficult. Let's see.
Zoom usually puts a green and red bar on the top of the screen with the option to stop sharing.
The problem is that it is not opening here. Let's see if I can go now. It was disconnected the presentation?
Yes.
yes.
Yeah, let's try again. Now I can see you moving. Appear?
Yes.
See if it is
Yes.
moving now.
Yes. Now it's
Yes.
It's
working.
move.
Yes.
Yeah, that is the second slide. That is why I talked about it before, that what is produced in the water, basic aspect of the exopolysaccharides, and the objective that it was to convert produced water waste in high value bacterial metabolic. Move it? It
Yes.
is move. Yeah. Then the objectives of the studies were assessing the chemical changes in APS grown in dialyzed produced water and distilled water to evaluate the impact of red lasers and LED on the EPS biochemistry, and also to use the 10464 nanometer Raman spectroscopy and PCA structural fingerprints. I will clarify later. The bacterial we use, it was the Leotia amnesia. We have these groups that is EPS produced in distilled water, EPS produced in dialyzed produced water, EPS produced and distilled produced water plus laser irradiation, and EPS produced on distilled product water and lead irradiated. We have prepared the growth curve of the microorganism, and we determined 2 points of irradiation. The first one was applied 90 hours post growth, that is in the log phase. You see here, that is the point that the bacteria start growing and maintain constant, and also 12 hours in the stationary phase to modulate the metabolism of the bacteria. We use a Raman spectrometer at 1064 nanometer that is very useful in this case of using in fluorescent materials such as biopolymers. And then we were able to see in the graph the results of the Raman spectroscopy of the different groups. We also carried out a spectral analysis for the identification of specific saccharides and acyl groups vibrations. We carry out a multivariate analysis to discriminate the biochemical signature between the groups. On the image, you see the scores that we used and the principal components found in each of the groups. The component 1, that is the strongest one, until the 5, which was the less strong, because after that we lose resolution of the study. So the first thing we did was to determine the chemical profiling of the exopolysaccharide. We detected on the backbone of the polymer the fucose, glucose, galactose, and mannose according to these spectra that are patterns for pure saccharides. We also found acyl groups by the presence of prominent bands for acetyl and pyruvyl groups, as seen on the right-hand side. And what the importance of these acyl groups? They determine the polymer's charge and ion binding capacity. that will influence on the future use. What we found on this study, when the photostimulation effect was detected, as we found a significant decrease in pyruvylation and increase in the mannose content. We also have a structural shift of succinyl groups, mainly carboxylate, that were identified only in irradiated groups. The influence of produced water, they favored the incorporation of the mannose, enhancing structural complexity of the biopolymer. It's well known that laser light targets cytochrome C oxidase that boost ATP availability on the microorganism. We also verified that acetyltransferase activity, they are likely to modulate enzymes responsible for pyruvate succinate substitution. So we see the mechanisms: biosynthesis, growth, exopolysaccharide production. And the effect of the light, we change the biosynthesis, the growth, and APS production. So what it means that external stimuli can be turned to engineer exopolysaccharide with specific industrial properties. The use of Raman spectroscopy showed a robust, non-invasive tool for real-time biopolymer monitoring. Environmental and light synergy. Both factors qualitatively redefine the APS chemical identity. Future outlook, potential for large-scale biopolymer tailoring using in the oil industry waste. So we use a byproduct of the petrol industry, that is the produced water. We use as microorganism the Leotia amnesia. We generated high-value biopolymers, as you see ahead. The importance of this initial study was that when we checked this polymer that it was initially tailored for the petrol industry, we decided to further evaluate it. And then we already carried out laboratory testing that showed that this biopolymer is biocompatible. That is a paper that I'm finishing to write. And also we did animal study to see how it works in vivo. Also, the results showed that the biopolymer is biocompatible. It can be used in human. For these tests, we used only the APS produced in distilled water, of course, because the composition of the produced water, it may have iron, metals in it that will be completely incompatible to use in human. But the results are very, very nice. The Professor Jean Nunes which also part of the ENCT, is doing the histological study with my students for there. And the possibility of using now in humans is well founded. And then we'll move forward now for deposit a patent for its use on humans as a biomaterial and also as a possible carrier for photodynamic therapy, using it as an encapsulated agent. This work was realized at the Federal University of Bahia, UFBA, in association with the Polytechnic Institute of Bragança in Portugal, através, with the CIMO and SUSTEC labs. And we are very much excited with the results, especially for the possibility of using in human, not only on the petrol industry. It may be used on both. Thank you very much.
Thank you, Professor Pinheiro. And
I'm
it
changing.
was
I'm
an
shifting here
interesting.
the screen.
Hi, can you see us now?
Now, yeah, now I'm back.
Okay, cool. So it was nice to see a new frontier that you can use photonics or biophotonics applied with potential use for dentistry. And I would before I open for more questions, I have 2 points that
Yeah,
I'd like to ask you. Yeah, you
yes,
told us that you use this distilled water now, but in a potential pitfall for the produced water from this oil extraction would be the contaminants.
yes.
So, what are you already working with protocols to remove these heavy metals?
The main problem is that there is a large number of contaminants, and then it's very difficult to find one technique to remove all of them, because we have iron, we have carbonate, we have magnesium, we have what else? Manganese, and
And
it's
it
very
may
difficult.
vary according to the source of oil, because I guess the which well might have a different composition, so it's a big.
Yes, and it's also different if you use the produced water. I didn't went too far on this matter, because you have the well on the earth, on the soil, and also you have the one on marine deposits like the
Mhm.
pre-salt. And
Mhm.
so the composition, even the composition of the oil differs. And then it is, let's see, tricky to try to clean, because they try
Mhm.
to clean using all the methods, and it are not effective at all. This is you notice that I use not the pure water. I had a
Mhm.
dialysis on this to try to clean a bit. I use a dialyzed produced water, not the raw one, that it's much more dirty, let's put that way. And we also have on this water that came out from the well, different microorganisms, including microorganisms that are not present normally on our environment, then this is very, very thing. It's this petrol industry is a bit crazy. I study with Professor Paulo Almeida, that is a microbiologist here, and we trace, we study using Pseudomonas aeruginosus, and we found it on the produced water. And interestingly, when we did the sequencing of these Pseudomonas, it was found that this species, which was found in a well in Sergipe, that we use to collect the produced water, it was only found on the north of Europe. So how can a bacteria, which is present only in the
Here,
North Sea, appear in a
here.
well here? And we
It's interesting.
did the sequence 3 times, and the result was the same. And then until now, we could not say how it happened.
Yeah, it's
Probably could
interesting.
be
Yeah.
something before we separate
Yeah,
the
before
continent.
Pangea. Yeah, okay.
Yeah.
So then it comes another question because these bacteria they might live in a really high pressure environment. Do you think these biopolymers that you are extracting and purifying, they might have some physical properties that are different that might be used for hydrogels, for instance? They might have a
Yeah.
higher melting temperature or higher rheology
We
properties.
we did much testing with it. The physical characterization, we have an increased thermoplastic activity. Is also possible to be converted in nanoparticles that can be used for instance, there is one very interesting thing on these polymers specifically is that we found a higher amount of fucose that is a very expensive sugar, and there is just one group in Portugal which produce fucose patented. There is one, we don't know even which was the bacteria because they use a code that is them. We don't know the bacteria they use, but they were the only one who produced it in the industrial scale. There is no mention, but we know that. And I don't know, have idea how the amount of fucose they have in their material. I it's industrial secret. They do not let us know, but it's very interesting because the fucose has been used worldwide in the produce of milk, even simulated breast milk, and also in the cosmetic industry. This is why he is valuable. And the most important thing that it was a second finding was the biocompatibility of the polymer. Is completely biocompatible. We test in the lab of immunology. It was fine, and then when I have the result of this, then we move it to the animal study that it was already concluded. Jean is preparing the final report to send to me. But from the results, we see that no problems in using it in humans.
Interesting. Yes, congratulations, really high technology. So I guess you might have a partnership with Petrobras for this kind of research, probably.
We had the initial study. It was in a project of Professor Paulo, that was supported by uh, politically, let's say that, not by Petrobras, but for Petrogal, that
Okay.
is the petrol
Oh,
industry in
okay.
Portugal.
Okay.
Because Brazil, there is a lot of politics involved. Then the project was referended by the National Agency of Petrol, and it was funded during 5 years by the Petrogal. And then it was a very big project that we work in different situations, even to check the biopolymers, produced water, uh, refueling or depollution of the fields, because the water, at the end of the day, it remains in tanks. When there is no more use, or you see that the well is dead. When the well is dead, when the cost of production and the amount of removed oil is not economical viable, and then they just shut down and left beside the residues, and that is a
Understood.
major a major product. And in Brazil, we have very old oil fields that are already locked. We are now, Professor Paulo is doing a very interesting project now, try to revive these wells, because now most of our extraction is on the sea,
Yes.
not on the land.
Mm.
And these
Yes.
dead wells are mostly on the continent, not in the sea. Other thing that we also found is that the bacterial colonies present in the pre-salt uh, water, or even petrol, are very different from the ones we see in the continental ones. There are some that are present in both, but there are some that are present only on the pre-salt region. We assume that is because of the deep depth of the field. So
Yeah.
it's very interesting. It's association with the people there. I remember once I went for a meeting with the people in Petrogol to present the preliminary results. And then Paulo presented to the top guys of the
Mm-hmm.
Petrogol here in Brazil. And then we had a talk, explain everything. And then one guy said, "Well, I like it very much, but what are your formation?" And then Professor Paulo said, "I am a veterinary med," and look at me, and I am a dentist. So they didn't understand what we are doing there.
Yeah. Wow. I don't know. Is there any more question for Professor Pinheiro?
There is, Marcelo, another very interesting derivative of this study is that we are also producing another, not a polymer, but amnolipid produced by Pseudomonas aeruginosus. And these amnolipids are very also used in industry. We are using it as a encapsulation agent for photodynamic therapy, either in fungi or bacteria. We use it to produce the nanocarrier for the photosensitizer, and it worked very well. That it was also derivative from this study.
Interesting. Yeah. So we need to maybe put in contact with people that are developing this photosensitizer. That would be nice. So congratulations. It was a really interesting topic, and uh, I guess we don't have any more questions right now. Is there anyone?
Marcelo, I don't have questions. I just want to thank Professor Pinheiro for sharing all this knowledge with us. And do you remember when we talked with Doctor the physic Doctor I forgot the name from Cividentos recently, Marcelo, and
Vladimir?
Vladimir, Dr. Vladimir. Maybe Vladimir and Professor Pinheiro could talk more because he have been developing different ways to try to reduce bacteria and analyze this kind of bacteria. He's working with Mycobacterium leprae. So
Mm.
Pinheiro, we can we can talk a little bit more after. He's
Yeah, we
also
have
using Raman.
Put in touch. Yeah.
in Salvador. We have a very problem, very big problem in dealing with microorganisms. Believe it or not, we have just one NB3 lab in the whole state. So our
Yeah.
capacity to handle some kind of microorganism is very limited. We have only the Lacene linked to the Fiocruz, which has NB3 facilities. The maximum we have at the university are NB2 labs.
Yeah, sometimes it's not easy. But thank you so
Yeah.
Yeah.
much,
Thank
Professor
you very much
Pinheiro.
for the opportunity. I
Thank you.
wish you a wonderful weekend and see you next time.
Thank you very much.
Bye bye.
Bye bye.
Bye.